to yet another Lifespan.io Journal Club. Dr. Oliver Medvedik here from New York City. We've got a full roster of people joining us. We've taken a little bit of hiatus because of because of illnesses and whatnot, but we are back refreshed and ready to go as ever. So once again, we've got yet another recent journal article in the field of biogerontology, longevity science, aging research, call it what you will. And let's see, let me share my screen and let's see what's up, what's going on here. What is this? It's a Nature Aging article. Does everybody see this paper on shared? Yes, participants can now see your application. My computer tells me. Okay, by Paula Juricic. Usually there's a stress on one of the C's. It's a Slavic last name. Don't know exactly how to pronounce that. Et al. Lots of authors here. And this is a mouse in Drosophila paper, mainly Drosophila and a little bit of mouse. And it's from the Max Planck Institute and along with a bunch of other institutes. And the title is Long Lasting Protection from Brief Rapamycin Treatment in Early Adulthood by Persistently Increased Intestinal Autophagy. So there's quite a bit to unpack here. So rapamycin has been known to have many geroprotective effects, beneficial effects, but there are problems with chronic treatment. One of the issues that was raised here is, I believe, a loss of insulin sensitivity over time. There's probably some other issues. So the question they're trying to address in this paper is basically how briefly can you give an organism rapamycin and still have protective effects, still have beneficial effects with regards to lifespan, longevity, and other beneficial effects. So that's what they're testing. So a little bit of caveats here, a little background. They use primarily or only female Drosophila because I believe the rapamycin treatments in general don't work as well or at all in male Drosophila. And they also are doing this in female mice. So, take that caveat there. So that's the background. Here's my highlights here. And short-term treatment, what they mean is chronic, meaning the lifespan of the entire fly, right? So they're dosing it with rapamycin, or during certain periods of time. So either later just give you a little background on the mice lifespan. I believe here they're living up to upwards of 100 days, and it ranges between different strains. So they're either dosing them later on in their lives, which were earlier studies had done, days 30 to 100, chronic, which is the dark black lines throughout their lives. But one thing that they've discovered in this paper is that you can dose them for much shorter periods of time early on in their lives. So they do days 15 to 30, and next figure they'll show days 1 through 15. Any quick questions here? So these are the lifespan curves. And what you're seeing here is basically, chronic administration, meaning rapamycin being given to the flies throughout. The colored lines here, the days that the rapamycin is being given, and then the negative control, no rapamycin treatment. So you could see that there's advantages to giving it earlier on. So chronic, you have noticeable increases. This tends to peter out as you basically give rapamycin much later in the stages of the fly life cycle. So days 60 to 100, you really don't see a negligible effect here, if anything at all. Days 1 through 30, you have quite an extensive effect. A little bit less days 15 to 30, but notice here that days 1 through 15 was sufficient. This is now figure 2, sufficient to basically have this prominent lifespan extension that's equivalent to what they consider to be chronic administration. So days 1 through 15 is basically, they administered it, I believe, as soon as the flies got out of their larval stage, which they call eclosion, somewhere here in the. So on day 3 following eclosion. So pretty early on, I don't know if how much of a comparison you can make this to humans, but I guess I'm going to stop sharing here for a little while, right? So it's brief, relative speaking, right? But I guess um, that's like still dosing somebody for the first 15 years of their life or so, right? And so I'd be curious to see how brief can brief get and how early can it get, right? I didn't look at the exact dosages of the rapamycin, so I'm kind of intrigued, if you can play around with this. Is an even briefer, higher dose of rapamycin possible? I'm not sure, right? So this really offers some tantalizing questions here. So anybody have any comments?
Yeah, Oliver, do you know what we knew about rapamycin dosing schedules before this paper? Like, I feel like it's common
Yeah,
knowledge that if you're just on
yeah,
rapamycin chronically, it's bad news.
yeah.
But I'm wondering if any studies comparable to this has been done. Like, it's used for other indications in humans, so
Yeah.
I'm wondering. Yeah, I kind of wish I knew more the backdrop of dosing schedules for this drug.
Yeah, I don't. Um, yeah, it's not one of those unlike metformin long term type of deals. So I'm not really sure what the optimal dosing schedule is for rapamycin for humans. Yeah. Um, but yeah, be interesting to see how low it can get. So one thing that they didn't do in this paper, so they did the fly lifespans, and I know folks always, we always talk about lifespan analysis as being kind of the gold standard. Um, that's not something they did. So I'm just going to kind of jump to the end here and then go back to the figures. That's not something they did with the mice. They did that with the flies, but they primarily the bulk of this paper now looks at gut health. So this is going to be the focus. And what the authors will show is increased autophagy and signs of autophagy in basically the intestine of the fly, and also comparable effects in the mice as well. So we do see these these improvements. Of course, there's a lot of other systems to look at in the organisms as well, other than the intestinal effects as well. And one other thing I'll just mention before to keep in mind as I bounce back into this paper is that the short-term dosing here, a lot of the effects that they see, obviously the lifespan is extended all the way through adulthood, so the maximal lifespan is increased as well, and we see a lot of these persistent effects, as they call it, that are beneficial with the shorter term versus chronic. So even if they take the administration away, you still, like 30 or 40 days later, you still see these beneficial effects. The one thing the authors didn't do, which obviously when you see something like temporarily administered and then you have a persistent long-term effect, I immediately say epigenome, right? Something is changed in the epigenome, because they do see, and it's in their supplemental data, so we won't look at it, but they do see histone changes, right? Changes, and so so if you get these persistent effects, and presumably a lot of changes in gene expression profiles, which they looked at some, but not a lot, I think. Then I think interesting to see what a short-term, maybe high dose of rapamycin would do to the epigenome and how stable that would be. So I'm just going to toss it out there because I don't think they talked about. That's one thing they didn't talk about in this paper is any potential epigenetic changes. I read it like twice. I don't think there's a lot of stuff in here that I'm not familiar with, and a lot of figures, but I don't recall seeing the E word in there, epigenome, anywhere. Okay, go back to. Okay. So we have this, relative short-term chronic, relative short-term treatment having beneficial effects. And now what do? They look at intestinal function, as I mentioned. So the short So what do they do for epigenetic function? A variety of different kind of immunohistochemistry cross sections and GFP expression. And so what they're looking at in these figures, C, D, E, F, and so on. So figures, what are we looking at here? So this is pH3 positive cells, which are phosphohistone 3 labeled cells, which are indicative of, I believe, cell division. I think it also gets labeled with regards to damage, but it's an indicator of cell division. So the older the cells get, there's more division occurring because I guess damaged cells have to be replaced. And there's less of that when you have chronic rapamycin treatment and day 60 after after administration of rapamycin from days 1 through 15. So this is their pH3 phosphohistone 3 positive cell count over, I guess, a certain region of the gut, right? And this is the numbers right here, chronic days 1 through 15. They also look at turnover rate. So they have this system here, this transgenic system, where it's basically a heat-inducible GFP tag that marks, I believe, intestinal progenitor cells controlled. There's a whole heck of a lot of them, and rapachronic and rapid days 1 through 15, you start to notice less of this turnover, which is indicative, according to this paper, of less damage occurring. And so on and so forth. What else do we have here? We have here diphosphorylated ERK here, which is indicative of damage as well. Those levels go down, so that's a protein. And they also look at what they call intestinal cell dysplasia, which is an aberrant shape of cells. I guess they get bigger over time. And they have this GFP resile, which I believe is like it's a membrane marker. So it's basically you can see the size of the cells, and the dysplasia, which is the size of the cells, the length of the cells, goes down. And they also do this kind of classic gut analysis, which uses a blue dye, this blue food color, which when you administer it to flies, when they eat it, basically, it will leak out of their guts if they get older. So as we get older, the connections between your cells tend to weaken. So these are your extracellular connections. So you basically have leakage between cells. So the flies turn blue. So those numbers go down as well, indicating that there's better gut stability, better gut integrity. Anyway, I'm going to stop sharing here. This is figure 2. And we got hands up. So, yeah. Oren?
Yeah, I think with regard to the cell dysplasia, maybe I got it wrong, but I thought that maybe they meant that like the cells, the intestinal cells, the intestinal epithelial cells, instead of being a single layer of cells, it's like more than one cell. Like when you see what you see in the control is that they are not only in a single cell, in a single layer, just near the lumen, but you have like more than 2 layers. I think that's what they meant with the dysplasia, but I'm not sure.
Yeah, I'm not 100% sure either. I looked it up. Basically, it's a variety of histological interpretations depending on which tissue you look at when they refer to dysplasia. So it's basically an aberrant morphology, if you will. So maybe you're right. Maybe it could be multiple layers. So you could sort of like a pre-tumor, if you will. Sort of you start having cells that are kind of losing contact inhibition, maybe. Could be. John.
Yeah, I suppose it's all histone stuff at the end of the day. I just so you know, and I don't mind how much I talk to you about this, but I happen to participate in the Rapamycin news forum, so I have used Rapamycin once and Hence, I do have a certain amount of knowledge about the variations. There's no real certainty people have about how to dose on rapamycin, and whether, in fact, at the age of 62, as I am, I'm too old to bother about it. I've taken it once. I intend taking it probably at a rate of sort of every 2 months or every 3 months. The dosage I haven't worked out yet, but the point where if you look at the gut issue, or whatever it may be, it doesn't really matter. It's all a question of mitochondrial efficiency affecting the histone through the cytosolic metabolism and the availability of acetyl-CoA, as I see it. So, that's how it appears to all hang together. And so, if your histone is short of acetyl-CoA, if the nucleus is a sort of acetyl-CoA and certain genes are not, certain parts of the histone are not acetylated, it doesn't produce certain genes, certain proteins rather, because the genes are not actually being not functional. And that seems, the paper on osteoporosis, which first came out with that approach, it just seems to be consistent through lots of things. Everything's consistent with cells as they deteriorate, as they become senescent or de-differentiate in some form, or fail to differentiate. They fail to produce certain proteins. And if it's epithelial cells in the gut, that's there. If it's in the vascular system, it's there. Wherever it may be, it's a similar thing. I don't mind talking more about that, but I don't want to sort of, if you've got questions about what people do about rapamycin, I don't mind answering them.
Yeah. Well, definitely, we're going to keep you on speed dial for questions about dosing, at least human dosing, since I've never taken it or used it in that capacity. And that's something else, when I jump back in here, that the authors didn't discuss, is that mitochondrial connection. So there are definitely a lot of gaps here that the data is really intriguing.
Autophagy is really about mitochondria, isn't it? It's sort of like about making mitochondria more efficient.
Yeah. Well,
right?
Yes.
So they talk about autophagy. One has to assume that follows through to the question: what's the key thing that autophagy is doing to the cells? It's making them more efficient and getting rid of rubbishy mitochondria.
Exactly. But that's something that they haven't looked at, which probably they should look at. So speaking of autophagy, there's figure 3. So they are looking at different markers or indicators of increased autophagic flux, if you will. So what are they looking at here? So lyso tracker, it's an indicator for, lysosomes. Is that correct here? Yeah. And they're looking at, all right, so a little bit more to unpack here. They're looking at expression of a whole bunch of different markers here that are indicators of autophagy. They're looking at, so basically, I'm just trying to remind myself. It's just, so, what are they looking at here? Cyto-ID and lyso-tracker dye. So cyto-ID specifically labels autophagosomes, and lyso-tracker states autolysosomes, and an increased ratio of autolysosomes to autophagosomes, reading this here, indicates increased autophagic flux. So I had to wrap my head around that here. So I guess as you have autophagosomes form, that they need to then fuse with autolysosomes, and if you have more autolysosomes, you have an increased, if I'm getting that right, an increased autophagic flux, and that's their metric. And let's see what the data shows. So they have lyso-tracker punctae. So this is figure A, and cyto-ID punctae. And this is the ratio that they're referring to here. So here's the staining of the cells. Here's the data down here. Control is basically, well, just no administration of rapamycin, rapamycin chronic, rapamycin days 1 through 15. So now I think the rest of the paper, they're now focusing on short duration of rapamycin, days 1 through 15. So you have an increase in lyso-tracker punctae. Here they're saying that cyto ID punctae is equivalent. So NS being not significant. I won't argue with this. It looks like might be a drop here, but according to their statistical analysis, those are not significant. Although lysotracker punctae, which is indicative of autolysosomes, goes up both with rapachronic and rapid days 1 through 15, and they're stating that increase in the autolysosome punctae, which are these the red points here, the red stains with the dye, versus no change in the cyto ID punctae, is indicative of increased autophagic flux. So I've never used this type of assay or this type of ratio to measure autophagic flux. So if anybody else is an expert and can comment on that, feel free. But that's the interpretation here, is that this is suggestive of increased autophagic flux. Okay, we'll move on to figure B here. So now they're looking at proteins that so biogenesis, which I believe is also Ref2P, which is P62 receptor for autophagosomes as well. And they also look at ATG1, which is an autophagy inducer in flies. And yeah, this gave me a little bit trouble interpreting it because these levels go down. So when you have chronic administration of rapamycin and also days 1 through 15. So, the take-home message, right, from 30,000 feet is that chronic in days 1 through 15 gives equivalent results. But this is the interpretation of this is that this is an increase in autophagy. And I'm just trying to figure out how in decreased levels of that is an increase in autophagy. Because ATG, if I've been reading it correctly, so it's responsible for basal biogenesis. So does that mean that the cells in general are healthier because they're undergoing less autophagy? If anybody wants to comment on that. Yeah, Oren.
I just have a guess. I have no idea. Maybe it's like there is some post-translational modification, and I don't know. No, it still should be the same amount.
Yeah.
The same weight. Yeah. So no idea. I will look. I am looking in the paper to find it. Maybe I'll find something.
Yeah, it's so they mention it. Oh, where they mention here? So they mention it. This is 2. This is 3. So we talked about the ratio here of autolysosomes and autophagosomes, equals of intestinal non-lipidated and lipidated forms of ATG8 protein and Drosophila P62 homolog P, and they stayed low. And of course, I'm just going to toss this out there. There was a tissue-specific response. So for whatever reason, they focused also on fly heads. So none of this was apparent in the heads of flies, which I guess, you're going to have most of your central nervous system located, as well as the fly eyeballs. There's a lot going on in the fly head, right? But so it's not happening in fly head, but happening in the intestines. So if the levels are equivalent to chronic in days 1 through 15, which is good. I'm just trying to connect this in my head to ATG8 and how a decreased level would actually indicate an increase in autophagy because it's from what I read, it's responsible for autophagy biogenesis. Yeah, Warren. Yes.
So I just wanted to say that I wrote in the chat the quote when they say that this is indicative of persistent autophagy. Maybe this protein is consumed. I don't know.
Yeah, perhaps. Well, if cargo receptor for autophagy, so P6, so I can see how those levels are down because if it's a cargo receptor, then it's going to get ground up and you're going to have less levels, right? Ref2P. I'm just kind of logically thinking this through. Anyway, those levels match what happens chronically. And when they look at the head, day 25, they see no such changes, right? So the levels they saw in these autophagy markers are not altered in rapamycin chronic or rapamycin days 1 through 15. I'm not a fly guy, so I don't really know. I guess maybe this is common, right? If you're looking at the intestines of a fly and then you just default to the head and take a look at versus the wing. Well, it's probably nothing in the wings, but I guess the limbs, right? So yeah, if anybody here is a fly expert and wants to fill me in on more of the logic on just deciding to kind of look at the head of the fly and whether or not that's just arbitrary or there's a reason for it, a deeper fly reason, I'm all ears. Okay, been told when I turn my head, the sound volume drops way down. Probably something to do with the laptop and the sophisticated AI system that is monitoring the placement of my head and is just throwing something off here. Okay. Sure. We're all being monitored. Okay, so you have induced lasting autophagy activation. So now they want to see, what is responsible for this induced autophagy activation. And they have some transgenic flies. And one of the things, so what do they do here? So they do that later, which is the gene ontology analysis. So what they do is they look at ATG5, and ATG5 is also an autophagy inducer. Let's see if there's anything more specific information on ATG5. So I'm not familiar with it. So this is figure 5. Nope. Should have boned up more on my fly autophagy pathways. It's just many autophagy pathways. So suffice it to say, ATG5 is an autophagy inducer. So they want to see if they can get comparable effects by knocking down ATG5 using RNAi or overexpressing ATG5. So interestingly, they have a system here. So they're focusing on the gut again. I think they moved on from the head right now. They're focusing on the gut again, but they're able to express this in the enterocytes, which are the intestinal absorptive cells, the columnar epithelial cells, nuclear, columnar, can't pronounce it now. So those are the cells that basically produce like the digestive enzymes. So I believe those are well differentiated versus intestinal stem cells, which are less differentiated. And they have these fly strains where they basically can induce and knock down ATG5, which is an autophagy inducer. And they do this in enterocytes and also intestinal stem cells. And I believe the take-home message here is that, and I'm not sure the logic behind looking at the two different cell types, because clearly there's a big difference between a differentiated cell and an intestinal stem cell. Yeah, Oren?
Yeah, I just wanted to say that I think that they use ATG5 as like an example for a protein which is required for autophagy.
Mm-hmm.
So they knock it down. So it's like, so they don't use it like an inducer. I mean, it's like part of a complex which is required to elongate the phagophore, according to Wikipedia. So that's all.
Basically, the earliest step for autophagy induction. Correct? Is that better? So basically, we're looking at autophagy induction in the sense of the initiation of autophagy, sort of like it's one of the initiators. So the question here is, what'll happen to lifespan if we block that, right? So we're basically blocking autophagy using this RNAi system to knock it down. They haven't done the overexpression here. Yeah, this is just knocking it down. That's the next figure, I believe. And whether or not blocking it is going to have an effect in one cell type versus another. And as we can see here, the effect is pronounced in enterocytes, but not intestinal stem cells. So if you have rapamycin treatment, both chronic and so what do we have here? Both, both chronic and short term. No rapamycin. So let's make sure I'm. And the inducing agent is RU. So no rapamycin. I'm surprised that if you knock down the autophagy that you don't get a decrease in lifespan. That's what I'm reading this correctly. Anyway, you don't see a decrease in lifespan, right? I mean, and I'm not sure if they commented about that. Now that I think about this. So determine whether direct genetic activation of autophagy was sufficient to mimic the memory of rapamycin in the absence of the drug. Sorry, that's the next figure. We're still on figure 4. There he is. So failed to increase lifespan specifically in enterocytes, but not ISCs. And it abrogated the protection of gut barrier function that's later on, so they're looking at the gut barrier function here, proportion of blue flies, right? And this is, they also do, this is actually, interesting. They do chronic knockdown of ATG5 using RNAi, and then it's now so UAS is basically they can switch on RNAi later. So both or switch it on for a short period of time, and then switch it back off again. So so this is both chronic. Okay, sorry, I got a little confused here. So this is both chronic administration of RNAi. And this is basically having ATG5 shut off for a brief window, comparable to this kind of brief window of, I believe. No, I'm sorry, I was reading that wrong. I thought that must have been the next figure. They're doing a lot of switching back and forth. But anyway, knocking down ATG5 doesn't seem to lower the lifespan. I don't know why, but it does block the lifespan extension if you have either chronic or long-term expression or long-term administration of rapamycin in enterocytes primarily. Anybody care to comment on that? Yeah, Warren.
Yeah, I would just guess that because they only knocked down the autophagy in the intestines, it's not such a big deal. I mean, maybe in the brain it would have been much worse, although it's a fly, so I'm not sure.
Yeah.
I guess in mammals, for sure, in the brain, if you knock down autophagy, you're in trouble. But yeah, I mean, I guess even if autophagy is like a hallmark of aging or loss of autophagy, still it's like it's not completely universal. It depends on the cell type. Yeah, so it's still.
Well, yeah, I mean, but blocking it in it's certainly sufficient to block the lifespan extension by rapamycin if you knock down ATG5 in the enterocytes. So it's certainly that location is severe enough to induce that blockage. Yeah.
Yeah, I mean, it's their way. They're trying to find out how the rapamycin works, like what pathway, what is the pathway that makes the rapamycin like store this data in memory that you took rapamycin. So I guess that's what they are seeing here, that autophagy is required to make this memory thing.
Yeah, exactly. So if we look at, so this is what kind of confused me here. So E and F. So E and F is chronic and brief rapamycin reduced in proportion of blue flies in the control group, but not. But they also have. Let's see. There's E here. Let's see. So the memory. So speaking of memory here. So what's the difference between, so is ATG5 RNAi here, and then their UAS ATG5 system. So that's an inducible system. So ATG5 RNAi, is that turned on from the beginning of, because they have another system here. I'm just trying to make sure that, so E and F. So if we're talking about persistent memory effects and taking a look at whether or not so I just want to clarify and make sure that I'm reading this data correctly, that E is a long-term induction of ATG5 long-term suppression using RNAi of ATG5 and enterocyte-specific chronic and day 1 through 15 overexpression of ATG5 RNAi abrogated by both chronic and brief. Okay. Yeah. Okay. So in this case here, so the memory effect for intestinal integrity does happen. It is affected by brief switching on of ATG5 RNAi, which is in this figure F, and figure E is a persistent expression of RNAi. So rapamycin and RU486 is none. So that's the inducer to basically activate your transgenic ATG5 RNAi. Proportion of blue flies goes up at day 65. RU486 also. So it's day 65 chronic. Rapamycin goes down. So is this not? Yeah, Oren.
So maybe, I don't know, I will try to say it, you tell me whether I'm wrong. So I think E is both rapamycin and ATG5 RNAi. Both of them are chronic. And F is both of them are days 1 to 15. I don't know why exactly they chose to do it this way. So I don't know. Does this help somehow?
Well, then according to this, if I'm reading this right, hmm. Well, yeah. So let's see. So then it's yeah. So it's blocking the it's blocking the effects of the rapamycin. Yeah. Okay. Well, yeah. All right. So I guess that's I'm just trying to think if there's any more information I can get out of this that it's blocking the effect of rapamycin, both in chronic, like you said, and in short-term induction of rapamycin in both situations. I guess that's all we can really get out of that figure. That knocking down ATG5 is going to block the effect of rapamycin. I think that's all we can take home from that for intestinal integrity. Okay, just thought that there might be something else we can pull out of there, but I think that's it. So then the next is overexpression of ATG1, and overexpression. Both, I think, long-term and short-term duration overexpression of ATG1. So they have these transgenic ATG1 strains here. And let me see here, because I think they also have two different types. Sorry for the confusion here, because they also have a figure later where they're able to switch on something for a short term versus a long term here. I'm just wondering if the ATG1 is turned on long term. Okay, so let's see here. Again, we're looking at Ref2P, P62, which is an indicator of cargo protein for autophagosomes. So we have short-term duration here of rapamycin. And we have, let's see here, RU46 is basically an inducer for overexpression of ATG1. So we're not knocking it down, we're overexpressing it here. And what are we saying here? That we have no significant addition. So basically comparable levels of Ref2P go down with either overexpression of ATG1 or administration of rapamycin. We have now in enterocytes, we have rapamycin chronic and rapamycin day 1 through 15. Overexpression of ATG1, which is responsible for autophagosome formation in enterocytes. No rapamycin control. Have an extension here and see both in chronic and short term. And I guess they're showing here that having an overexpression of ATG1 has comparable effects if you overexpress it in gut enterocytes in lifespan extension as administration of rapamycin. And similar effects basically, supposedly, although when we look at this data here, this is again proportion of blue fly, so this is gut integrity. What are we seeing here? We're seeing well, they're saying these are not significant. So rapamycin obviously improves gut integrity. having overexpression of RU486 doesn't improve it, meaning overexpression of ATG1. So you have, well, seems slightly better with rapamycin. Yeah, Oren?
I wanted to say about the panels before, the survival curves, that this is really like ATG1 is an inducer of autophagy, and you see here that, like on the left, that only ATG1 does better than rapamycin, or maybe the same, but no, I just wanted to say that earlier when you said that knocking down autophagy didn't reduce the lifespan, so now I see, I mean, now it's weird that if you overexpress autophagy, you get a lifespan extension, but really if you knock it down, so you don't see anything, so it's really weird. So if earlier I said back that it doesn't, it's not surprising, I take it back.
That's okay. We have new data. That's how science moves. We change our minds based on additional data that we see. So yeah, so overexpressing, increasing, so that kind of makes sense that you would block these effects if you knock down proteins that are responsible for inducing autophagy. So I'm not sure why. And of course, if you overexpress proteins responsible for that, you get an increase, right? So the inverse. But why knocking it down also doesn't lead to further reduction in lifespan? I'm not sure. Is this peculiar to fruit flies, or is this also something that you would see in other organisms as well? The authors don't kind of mention that that kind of that effect, that why we don't get a decrease. I don't recall seeing that in the discussion either. So maybe this is common knowledge in the fruit fly lifespan autophagy circles, but I'm not sure. So anyway, so short term. Okay, so figure 5. So this is what I was saying. So. Okay, yeah. All right. So that was sort of the big deal here, is that this this expression of UAS ATG1, something that I'm mentioning here, it's also short-term treatment. So that's also kind of the big deal. So the overexpression here is short-term of ATG1 overexpression, right? So this is also, so that's kind of the one connection here. The one major connection to make is that the overexpression of this is also not chronic, but it's induced for a short period of time, comparable to rapamycin treatment, and that you have these long-lasting effects. So that's yeah. So let's see what they're looking at in figure 6. So they do here, so just justification for figure 6 is they're looking for additional factors that are responsible for regulators of this memory effect of rapamycin. So that's what they're referring it to, because you have short-term induction, days 1 through 15, and you have this persistent effect later, right? So the flies live longer. So they do a proteomics analysis, and they look at stuff that's basically, after rapamycin treatment, stuff that's enriched in various metabolic pathways. And they mention that they have proteins involved in branched chain amino acid, BCAA, carbohydrate metabolism, particularly lysosomal aminocytases. So you have an increase in LMAN5, lysosomal alpha manocytase. So they also check that with check the RNA levels. So then they wanted to test if knockdown of LMAN5 abolished it. Now, I looked at the extended data 5, and they mentioned like 5 or 6 things there. So I went into the extended data 5 again. I was like, maybe there's enzymes involved in, altering the epigenetic status of many genes, right? But it was kind of an abbreviated list. So I don't know how many they actually looked at here. So if anybody saw like a longer list, but I think it was like 4 or 5 things that were mentioned that were shown on extended data 5. So I would be curious to see, I would think that if you dose these animals with rapamycin, that you would have a lot of different changes happening in a lot of other different genes rather than 4 or 5. But that may have been just the ones that gave the strongest response, and that's what they focused on, and that's what they showed. So, and maybe those are the ones they focused on because they're also involved in autophagy, right? So LMN5 is lysosomal alpha-amidase 5, which breaks down complex sugars from glycoproteins.
Yeah, I think the reason that they didn't see like a lot of stuff is because they looked on day 25, like 10 days after they stopped the rapamycin treatment.
Mm-hmm.
So they are just looking at things that stay long term, like this memory effect.
Mm-hmm.
Yeah, I guess otherwise you would see all of the things that usually everyone sees when they use rapamycin. I guess this is something that many people have already done.
Yeah, it's just when you think of memory effect, I keep going defaulting to the epigenome because it's like you have a persistent expression of these proteins, right, for like the rest of the life of the organism. Something's keeping those levels up, right? Those mRNA levels have to be cranking out. Something must have changed in the genome to keep those. The flip side of that is that those proteins aren't being broken down as readily. But I can imagine that for a short period of time. I can't imagine it for that you have the same protein lingering for 50 years, right? I would think it would make more sense that you have more persistent expression happening. Michael.
Yeah, so I think the same thing. And when I read this paper, my biggest question is the persistent effect real? And is it mediated the way they think it is? Mm-hmm. And so here's an alternative explanation for their findings. They have a lot of follow-up at 10 days, like in their inducible constructs, where they show there's changes in various lysosomal proteins. Mm-hmm. They also have other long-term follow-ups. They have like 60-day follow-ups on intestinal permeability. They have their survival curves. Unless I'm missing something, they don't have the 60-day follow-ups for the lysosomal pathway or the autophagolytic pathway. Mm. In my mind, a simpler explanation for some of the findings here is you have a medium-term upregulation in like lysosomal activity, medium term being like 10 days, and maybe that's what has the persistent effect. That's a lot easier to swallow because, like, when you think about what rapamycin does, there's a lot of overlap. It's like it affects protein translation. It's kind of like a throttle for your ribosomes. There's a lot of overlap between rapamycin mechanisms and like fasting's mechanisms. And no one is saying like if you go on one fast, there's going to be effects for life, right? Like that would be that would be insane. So I'm really wondering if maybe there's a somewhat more modest interpretation of how this works. Maybe it's not persistent upregulation, unless there's some data in here that I'm missing that proves me wrong. Another key number to think about here is like in the intestine, at least in humans, your cell turnover is like 5 to 10 days. Mm-hmm. So there's a big difference between stuff that you see happening on the time span of 10 days and on the time span of a month. Because if you see an effect on the scale of a month, it's not even the same cells anymore. Mm. So that's really striking if it truly is a persistent effect.
Yeah. John?
Yeah, well, an alternative hypothesis, of course, is that through the autophagy, the senescent cell load is not increased as quickly, and therefore that has a memory effect because of the reinforcing effect of senescence.
I think that's a good explanation. So senescence is comparable because senolytic dosing is intermittent. If this isn't widely done, but Medvedik is something where you might have a drug that's you dose for one day and you're off for a month, and it kills senescent cells. And you don't have like month-long downregulation of BCL proteins by a brief dosage of navitoclax. What you do is you have lasting killing of senescent cells. So it's the protein level effects are very brief, but the tissue level effects are long. So you can imagine something similar happening here.
Yeah. Does Tova have a question? Or is that gesture supposed to be clapping? I think it's clapping. Applause. I have to figure out what the icons mean. But if you did want to say something, please feel free to jump in. Okay, so yeah, so now they're looking at the lysosomal alpha mannose 5 expression levels, and they're doing similar effects here, which is doing similar effects, similar experiments, I believe, in knocking it down and seeing if it's, mediates memory of autophagy, and according to the figure legend, it does. Persistent increase in Mailman-5 mediates memory of autophagy. And let me see, remind myself if they're doing short-term overexpression or long-term overexpression. Sorry. Shut my phone off. Let's see, overexpression. Okay, let's go back. Okay, so they're looking at LMAN5 for the reasons that we just mentioned earlier, and doing similar rounds of experiments to see what would happen. So they're using, again, transgenic system here to express RNAi to knock down LMAN5 levels. Let's see here, 10 days after switch, and 45 days after switch. So just trying to remind myself what the heck they're doing here with the duration of the switch of the LMAN5. So RU chronic, yes, okay. So RU chronic is basically chronic dosing of RU486 that is going to be switching on RNAi. And then they're looking at, so there's some typos here. This is supposed to be Rapa plus RU here. And then short-term duration of RU486 days 1 through 15. So they're looking at, let's take a look at here. So lyso tracker. So again, this is the punctate foci, which are indicative of, I believe, autophagic flux. Let's see. Control levels are down. RU chronic. What's going on here? So this is after. God, I'm having difficulty for some reason interpreting the figure that they have here. Looking at the legends here. So when are they actually switching on the RNAi? So rapamycin days 1 through 15. You have more lysotracker punctate. Rapamycin plus induction of RNAi. So you're basically knocking down L-mannose 5, lysosomal alpha mannosides 5 in days 1 through 15. So that's abrogating the effects of rapamycin. So basically, if you're knocking down this enzyme, which breaks down complex sugars from glycoproteins, and yeah, I don't know. Does anybody else can chime in on anything else that lysosomal alpha mannosides 5 does that break down complex sugars from glycoproteins? Because it seems to be, at least for the formation of these autolysosomes, critical for that. You seem to have less of these punctae. And I'm not sure why LMAN5 is going to be the kind of the linchpin to all this, but it turns out it is. So versus all the other enzymes that are in there actually also doing a lot of garbage disposal work. But that seems to be that came out of their analysis. That's where they're looking at it. They're looking at let's see where they pull this out here. Yeah, that was from their gene ontology term enrichment analysis. Also found an increase in lysosomal alpha amidase 5 mRNA levels. Hold on a second here. Okay, well, there you have it. LMAN5 is kind of the main player. I'm not sure if I should be surprised at that, but if anybody else is surprised, but there you go. And what else happens? So if you also knock down LMAN5, you look at other effects here. So they're looking at intestinal dysplasia, right? So this is again aberrant morphology, intestines. So DAPI staining here. So they're measuring dysplasia, the morphology in some way. You have a control. You have RU4086 treatment days 1 through 15. So you do have, and then rapamycin 1 through 15. So you have a decrease in dysplasia, and then again you have an increase in dysplasia. It seems you're blocking maybe even higher than average. You're blocking it by inducing L-mannose 5. Sorry, it's called L-man 5 RNAi. But you don't. Yeah, I'm not sure why you don't get an increase in dysplasia by knocking it out in the controls. That's kind of like. It's kind of like similar to what we saw earlier with the lifespan, or right with the ATG5 overexpression. Like you get an increase, but if you knock it down, you don't get a decrease. So if you knock down LMAN5, why don't you? Maybe I'm reading this wrong here, but you don't seem to get worse. Let's put it this way. But you do block the effects of rapamycin on having effects on dysplasia. Yeah.
Maybe it's like rapamycin is doing a lot of stuff. Some of it is good and some is bad. And now you block the good and you still get the bad. So I don't know, but yeah, it's weird. But maybe this intuitively explains it.
Yeah, I think we're gonna have to roll with that explanation for now. All right. Okay. So what are we doing here? So I guess this is persistent versus short-term effects. So you have, so now they're looking at, so what are we looking at here? 10 days after switch. Now we have overexpression. All right. Now we're not, so now we don't have RNAi, we have overexpression of LMAN5. So let's see what happens when we overexpress LMAN5. So control, so now we're looking at, the autolysosomes and what happens when we increase the levels. Are you chronic and are you days 1 through 15? So you get a similar increase. So now they're basically doing a short-term duration expression, overexpression of L-mannose 5. And overexpression decreases also dysplasia. So let's see if the numbers are about the same here. So if you have rapamycin, total length is 0.2 on this. Total length kind of goes down to a 0.2, 0.3. So comparable kind of decrease. So if you overexpress LMAN5, you can kind of get similar effects as to rapamycin short-term treatment. And what are they looking at here? So this is now, I believe, in combination with rapamycin. So what's the difference between lysotracker ponte and lysosomal intensity area? So F. I'm just going to say as an aside here that so they use tetracycline also to knock down the gut microbiome. I guess this is used in flies. Suffice it to say, giving flies tetracycline doesn't abrogate these effects. So I think they mention here, and I'm not sure how much you can get out of this. I mean, they didn't measure the microbiome. So there is an administration of tetracycline and then a claim being made, figure 6. See, we found that they were incorrect. These responses to rapamycin were unaffected by tetracycline treatment, suggesting that the intestinal microbiota did not play a role. So, okay. So I don't think it was directly measured, like what if there was an actual change in the microbiota, other than the statement that if you give them tetracycline. So, I guess there's an assumption here that your tetracycline is working and it's really wiping out your microbiota and it's not having an effect here. But the effect that they looked at was lysosomal intensity over area, which is another metric here. So levels of intestinal lysozyme are a proxy for secretory autophagy. So they mention here that studies show that lysosome associates secret. So secretory autophagy is an autophagy-based alternative secretion system that is activated in response to infection. And it's mediated by core autophagy proteins ATG5 and ATG16L1. So I'm not too familiar with secretory autophagy. I guess it's an autophagy where you have autophagosomes and then the cells just kind of vomit it out and goes into the intestine and, well, out where you would expect it to go. So I guess that's part of the one of the autophagy pathways for garbage disposal in the cell. So intestinal lysozyme. So that would be an enzyme that basically is found in, that's, breaks down, bacterial proteins, for example, carbohydrates, peptidoglycans, I believe, in the cell wall. So if you see increased intestinal lysosomes, that's an indicator of secretory autophagy. Lysosome intensity over area goes up if you have LMAN5 overexpression. And is this supposed to be significant? I don't know if it's a slight drop. I believe they show that, state that having LMAN5, let's see what we have. Oh, this is RNAi now. I'm getting confused. We've got RNAi and overexpression here. So control RU486 goes up days 1 through 15. Let's see what we have here. According to this, that's a not significant p-value. Rapamycin days 1 through 15. You have an increase in lysosomal intensity versus control. And that effect is abrogated to some slight degree. Say it's P008 by having RNAi for LMAN5. So I'm not sure exactly how how much of a role LMAN5 is playing for the secretory autophagy. Yes, Arn.
I just wanted to say that this is really weird. First of all, it seems like if you look at both controls, like F and G, the controls are pretty different, but I thought that they should be the same. I think it's the same experiment, and the controls are like here on F it's 5, the control, and on G it's 15. And then you have, on F you have overexpression of L1, and on G you have RNAi, and in both cases, I mean, okay, it's on F. Okay, on F it's RU days the darker one. On F, the darker one should be like the opposite of, right? The opposite of.
Sorry, you muted yourself. You're still muted, or
so
there.
So both of these should be like the opposite, right? Because one is overexpression on the left, and on the right it's RNAi, and both of them are increase the intensity relative to the control. So it's really weird.
Yeah. So yes, you're right about that. I'm just wondering if there's also an error here in labeling, but it shouldn't be. So you have so let's see. Well, they're saying that that's not a significant difference between. I mean, if we're looking at, if we're taking these numbers, we're just accepting the numbers, the p-values here, then they're saying that there's not quite a difference, even though there seems to be a trend between these two here, which is RNAi versus this and this, which is getting to be, the acceptable borderline of 95%, right? Actually better than that. Sorry, that's a P005 of much. So according to this, so this is greatly significant versus p-value of 0.064 between these two. If we're just going by those values here. Although I see what you say about the trend here. Okay. So it does seem to, LMAN5 does seem to have an effect, comparable in some ways to, short-term, days 1 through 15 duration administration of rapamycin. It's, you're having an effect, especially on the formation of these punctae that are autolysosomes, and the dysplasia and other effects as to rapamycin. And I don't know if again, if we're looking at the whole lifespan of these flies, I'm not sure if all of this could be accounted for. I'm not going to say LMAN5 is the master regulator for rapamycin lifespan extension in flies. Perhaps in autophagy, maybe in intestinal enterocytes, but that's again, that seems like a valid assumption to make based on that data. Okay, now they switch. So now we are switching to something completely different. Figure 7 and 8. We go to mice. And as I mentioned at the beginning, they don't look at lifespan of mice. They're basically looking at intestinal effects again. And they do this for, they do for these experiments though for quite a long period of time. So, I mean, it would have been interesting to see the survival curves, I think, because they say tissue collection at 12 months. So, mice live, well, it depends on the strain of mice, but what, 2 years for about a typical lab mouse? Maybe 3 is pushing it. So 12 months is, 12 months is like mouse middle age. So anyway, they're looking at a number of different factors here, which basically constitute the health of the gut of the mice. So here's some cross sections. Looks like EM microscopy here, and there are some junctions here. So all your cells are kind of welded together using proteins that are basically forming junctions that prevent things from basically leaking between cells, from the lumen of your gut to the rest of your body. And these morphological indicators here are I don't do gut junction analysis, so I'm going to have to take their word for the nature of these. That if you can spot the quality of a junction, intact, intermediate, damaged. If you look at basically how diffuse the junctions are. I'm not sure if they used a kind of a color-based assay as well, like they did with the flies. I think they did analysis of EM microscopy to look at the integrity of these junctions, these tight junctions. They also look at LBP, lipopolysaccharide binding protein levels, in the blood of the mice. So if you have leaky junctions, you get more bacteria kind of flowing around in the blood of the mice, and you have more of this LBP protein, right? So, so let's take a look at the data here. So LBP protein levels go up as mice get old. And here's a control. And, it's a slight decrease. Rapa chronic, Rapa months 3 to 6, no significant difference between Rapa chronic and Rapa, so this is a short-term duration treatment. And let's see, did they do that actually for the first? They do months 3 to 6. Could have sworn they also did this for like the first few like months of the mouse, kind of comparable to what they did with the flies. Treated mice to Rapamycin chronically or from 3 to 6 months of age. I'm wondering why I didn't do it months 1 through 3. Anyway, Warren.
I just guess that because of development in mammals, it's really different, and you can't just use rapamycin early. Just a guess.
Yeah, well, there must be some precedent. There must be a paper that dosed mice. Somebody must have tried giving rapamycin to mice when they're 1 month old and their heads exploded, and published that that's a bad idea. I'm being a little facetious there, but there should be some paper on that, right? Because I would think the logical thing would be to start it as early as possible, based on the fly data. But anyway, months 3 to 6 it is, short-term duration. But again, one thing that I'm curious to see how short-term and short-term could be, because in the short term is if we extrapolate to humans, right? So if a mouse lives 2 years, right, 3 months is still a considerable chunk of time in the mouse lifespan. So that's kind of like, if we're going to compare it to the flies as well, and just, we can make a very loose comparison to humans. It's kind of like dosing like throughout your entire adolescence or your entire early adulthood period. So it's a pretty decent chunk of time. It's certainly not like comparable to a month of rapamycin. More like 5 to 10 years of rapamycin, if not more. So anyway, just tossing that out there. So when we're talking about short term, it's a relative term here versus taking rapamycin throughout the entire lifespan of the organism. So okay, so LBP levels go down slightly, but again, the claim here is that it's significant, and the number of intact tight junctions goes down. I don't know why you got this slight increase here, but it's just not significant from 12 to 22 to 24 months. Oh, so they do take these mice all the way to like way late, right? So 24 months. And this improves. So rapamycin chronic and also the short-term 3 to 6 month treatment, has an improvement in tight junction stability. So that's the data there. Next, they look at specialized secretory cells. They also serve as an age for ISCs. So they secrete things, digestive enzymes, whatnot, into the lumen. And I think their morphology changes negatively as the the kind of the secretory granules, the lysozyme, which is secreted out. So this would be the lumen right here, and these are the cells right here, and this is the staining of lysozyme. They talk about diffuse and various degrees of diffusion, percentage total of Paneth cells. So this is a measure of basically, I think, the quality of the cells, if I'm reading this right. So if we scroll up to, let's see, I already looked at lipopolysaccharide binding protein. Let's see what we have here. So we also looked at tight junction morphology here. So lysozyme is normally efficiently packed, and abnormal lysozyme distribution, which is more diffuse in cells. So basically, I guess basically the secretory granules, the secretory vesicles are just kind of more abnormal. So let's see, this is a measurement of cell abnormality, kind of an intracellular version of the dysplasia that we kind of talked about earlier, if I'm interpreting that correctly. So if we, and this is their measurement. So percent total Paneth cells. So basically, more of this kind of white area, less of these numbers here, which constitute, I guess, the number of granules that you can count that are less diffuse, is better. So if we have more of these granules that are more kind of punctate and you could see them, then that's a measure. Again, I'm not familiar with this technique here, but supposedly 6 months after treatment, you have less of these diffuse granules in these Paneth cells. And the same thing applies to the density of these granules, which basically is packed with a variety of enzymes for secretion out into the gut lumen, right? So these hypodense granules, the number of these goes up with time, and chronic and short-term 3 months, 3 to 6 administration of rapamycin tends to decrease them, basically decrease in the hypodense granules, right? And finally, last, what they look at is short-term rapamycin exposure increases the number of granules positive for both lysozyme and which is a cargo receptor for autophagosomes in Paneth cells, and the number of mannose 2 B1 punctate. So that's I think that's the equivalent of L-5 in mice, if I'm not mistaken. So I think it's a lysozyme alpha-manidase 5 to the same degree as lifelong treatment in mice. So again, they're looking at so P62 lysosome granules per cell. So I believe this is indicative of autophagy. That goes up both in chronic and in short-term treatment of rapamycin. And also these mannose 2 B1 positive punctate go up as well. And I don't think they did, as they did with the Drosophila, I don't think they did any more genetic analysis to see if what is actually kind of blocking the effects of the short-term and the long-term rapamycin here. So yeah, that is the data. I'm not sharing here. So there was a lot of detailed assays, a lot of different assays being used, a lot of focus on the gut integrity, both the gut health, if you will. So Oren kindly put a link here in our chat to rapamycin treatment during development extends lifespan and health span of male mice and Daphnia magna. So it's not bad during development.
Yeah, so I take my words back again. So it's day 1 to 45. It's a Gladyshev lab paper. It's 2022.
Okay. More recent, or maybe did this come out after this paper, or just?
No, but I'm sure they were both prepared like a long time before that. So
yeah.
You can know. But maybe they talked. I don't know. Still, I don't know why they did it. Like why they chose 3 months to 6 months. I have no idea.
Okay. So yeah, there's a lot of questions in this paper, but I think for me the long-term questions on the short-term experiments here, short-term administration, is again, like I mentioned before, how high dosage and how short-term can you get, and these supposed persistent effects, what underlies them, and what is actually the critical point. I think LMAN 5 is way up there. I don't think LMAN. That's just my thinking here. I don't think that, at least for the lifespan extending effects that we see in general, in toto, for the flies, and perhaps for the mice, they didn't do the lifespan here for the mice, but I would gamble that it's not LMAN 5 being the critical master regulator. That it's probably something more fundamental to do with nuclear architecture or gene regulation, right? So kind of you want it at that level, and that you're gonna that the effects that we hear that are positive for the gut, that you're probably also going to see positive effects if you look at other tissues as well, and maybe autophagy isn't or the regulators of autophagy aren't necessarily underlying that for some of the other tissues. I'm not sure. They seem to, they looked at the head here, which they didn't see these changes. I guess there's a lot of stuff happening in the fly head, so it's not like you've purified neurons. So I don't know exactly what to. There's clearly no intestine in the fly head, at least that I'm aware of. So there's none of that tissue, but there's a lot of other stuff there as well. So I would think that you're going to have a kind of a broad spectrum effect with rapamycin on many tissues, and it's not all kind of coming out of the intestine and just LMAN 5, right? So anyway, that's my 2 cents. Is that these persistent effects are cool, and how short term can we get with these effects, and what really underlies it? Are there epigenetic changes, and how persistent are those changes? Is there a certain threshold that needs to be made, and can we quantify that, right? I think that would be kind of a cool experiment, is to do different rapamycin dosing regimens and take a look at methylation status and kind of plot a curve and see if you kind of hit a sweet spot for dosing, and see whether or not you can optimize this effect. So anyway, that's my comment on that.
And that's about all you got to say about that.
Yeah.
Well, okay, Forrest. Well, hopefully that's generated some entertainment for you all today. I certainly learned something about flies.
Mm-hmm.
And maybe a little bit about mice, although I like mice a little bit more than flies. Flies are a pest. But there you go. That's the gardener in me speaking. I don't like flies. Not fun. So I guess you'll be regaling us with another entertaining paper next month.
Yeah, unless anybody has some suggestions. I'm always open to suggestions. So I know. Yeah, I can't. I mean, I can always be picking papers, but please send submissions if there's something. Maybe the Gladyshev paper that you just mentioned, Oren. Take a look at that.
I might be able to send some things your way. There are some interesting stirrings. There's always interesting stirrings going on, but yeah, there's been some interesting papers.
Yeah, that one just came out September 16. Rapamycin treatment during development extends lifespan and health span of male mice and Daphnia magna, a different model organism that we have. I don't think we've ever done a paper on Daphnia, which is, I believe, a little aquatic freshwater,
like a water flea, isn't it?
Yeah. So it's a metazoan, and there's been some usage of it, I believe, in longevity studies recently. So not as much well known as all the other workhorses of molecular bio, C. elegans, et cetera, Drosophila. But yeah, maybe we can switch it up and talk about a completely different model organism and its suitability for biogerontology.
Yeah. Perhaps we can talk about a little bit about why people are using it as a model as well before we, as a preface, before we dive into the paper, because as you say, not that many people know about Daphnia. They're interesting, and there are some actually quite valid reasons as to why they're used. So yeah, we could probably explore that. So not just a tasty snack for your tropical fish, folks. I used to keep tropical fish, so I know. So not just a good quality type of live feed for your pets. They are also very useful in the lab, it seems. And I know that some companies have been looking at them and exploring using them as a model and actually tracking their movements with video cameras to see how it changes their behaviour, during a test. But anyway, let's not talk Daphnia now. Let's not get too carried away tonight, lads. Too much excitement. We'll save that for next time. Whoa, those come in pints. Maybe the Christmas special. We can do something festive, because that's not long away as well. So
no, but what is coming up short term is Halloween. So yeah. Everybody, those of you going out there, well, it's on a Monday this year, at least for us. So
it is. I know in previous years we've actually done a Halloween one. So as I always say, Halloween, whatever, the biggest scare story, the horror story is aging. I don't think that's that's really refutable, and maybe taxes, but yeah, aging is the true horror story. So I guess if you are celebrating Halloween next week, have a good one.
Yeah,
a lot of people like myself will be celebrating it in the coming weekend because people don't like it when you drink at work, and it's generally frowned upon, Michael. It's definitely it's not the done thing, maybe 20 years ago.
Especially if you operate heavy machinery or drive a school bus.
You say that, Oliver, but 20 years ago when I used to work in a steel plant, that's right, it was very customary. This was before the days of health and safety really came in the UK, and 12 o'clock lunchtime, people would just go across the road and they'd have 2, sometimes even 3 pints in the lunch hour and come back, and it's like, yeah, let's go and hammer some stuff and use a massive industrial press because that's not dangerous after 3 pints.
I like you how you put that, Steve. Back in the old days of 2002,
2002
everybody used to
was the
operate.
year it began.
Everybody used to. You're like 20 years ago. I'm like 20 years.
1989 was the year it began.
Back in
I was a
the
young
UK,
man.
all the nuclear power plant operators were loaded. That was back in the old days, in the early 2000s.
I don't know about the nuclear, but I do know that this went on at a steel mill and pressing plant that I worked at. So very common practice in the UK. We are a little bit more health and safety conscious now. It was a simpler time back then.
Pre-Facebook, yeah, it was a simpler.
Yeah, it was. It was actually. So
pre-iPhone too. Yeah. Pre-smartphone. 2007. Yeah.
I worked there before then, so yeah, it would have been that would have been back in the days of, if you want to date it, like the Nokia 3210. So yes, those days, the brick.
Yeah.
But
anyhow,
memory lane can wait.
We'll pick a paper that will be different. And as always, we try to mix it up, and we'll see what we'll have next month. Thanks, Oren, for that paper from the Gladys Chef Lab. I'll take a peek at that, and we'll see what we have. You read that paper, Oren?
No, I haven't. So I don't know whether
I don't know either. So I'll open it up and take a look.
No spoilers, though, right, Oliver. So no spoilers until next time. We will be back, unless obviously we all come down with the dreaded COVID, which was unfortunately what happened last time. So that's not good. You know, at the end of a vacation, really not what you wanted. Not ideal. So, and then quite a few of us were actually ill elsewhere in the company. It was crazy. It was like we come back from holiday. Oh, we're ill. There were about 5 or 6 people ill. So, a lot of it around. Anyway, and on that note, stay safe. We will be in touch via the usual channels. Thanks to the heroes who support this and many of the other wonderful things that we do. Thank you very much to the heroes, the folks here, and the folks who are not here as well, but are still heroes to me. So, and if you'd like to learn more about Lifespan Heroes, visit our website. We are at lifespan.io/heroes. We'll see you next time.
All right.
Thanks for doing it.
Thank you. See you later next month.
Thank you.
Take care.